Hello, let's look at our second lesson in our trilogy of modes of inheritance of alleles. Our second lesson today is looking at the inheritance of incomplete dominant alleles. By the end of this lesson, you should be able to demonstrate your understanding of inheritance of incomplete dominant alleles using Punnett squares. But first, a review of last lesson, the complete dominant alleles. If you remember, an allele is a variant of a gene. For example, if the gene was hair, then the alleles might be curly or straight. A phenotype is the physical trait that is visible on the individual, for example, eye colour. If it's visible, it might be eye colour or hair colour. If it's measurable, it's something that you can't particularly see, but still occurs within the individual. It could be the production of a blood protein, for example. A genotype is the set of two alleles inherited from your parents, for example, AA, capital A, or capital H, lower h, and lower h, or lowercase t. In complete dominance, the letter used for the allele is taken from the dominant allele, and the dominant allele masks the expression of the recessive allele. For example, if brown hair is dominant and red hair is recessive, the allele is named after the brown hair allele, and so therefore the alleles would be, for brown hair would be the capital because the brown hair is dominant, capital B, and for red hair allele it would be with the lowercase b. So we've taken the lowercase b from the dominant allele, which is brown hair. In this lesson, we're looking at how the inheritance of alleles through incomplete dominance affects the phenotype and genotype of the offspring. Okay, incomplete dominance of alleles. So in incomplete dominance, the heterozygote offspring has a phenotype that is intermediate between the phenotypes of the two homozygous parents. Neither allele is dominant over the other, and they are both expressed in the individual. For this example, we've got a red snapdragon. Now, the red snapdragon has two alleles. Both of them are expressing the red pigment, so capital R, and we're crossing that with a white snapdragon who has two alleles for white pigment, capital W. So both of the parents are homozygotes. Excuse me. If they are to be crossed with each other, each of them only has one gamete to donate to their offspring, and so they have no choice but to donate either the big R in the case of the red snapdragon or the big W in case of the white snapdragon. And you can see that each of their offspring will have one of those alleles each, capital R and a capital W, and so therefore they'll have an intermediate phenotype different to both of the parents. So the RW combination would provide a pink colouring rather than a red or a white. All right, let's use a Punnett square to calculate these probabilities. This is going to be our first cross, and it's just the same as we did in the last slide. Both of our parents are homozygotes. The first one is a red homozygote. We've established that as parent number 1, and the second one is a white homozygote as parent number 2. And they have the same genotypes as the ones in the last slide. So I've put parent number 1 in our Punnett square down on the left-hand side, and the parent number 2 genotype on the top side of our Punnett square. And we can see using the Punnett square that all of our possible offspring will receive one allele from both parents, and so therefore they're going to get this intermediate genotype RW, and that's going to be both red and white, or give a completely unique phenotype in giving a pink colouring to all of those offspring. So down here I've got a little denotation saying F1 or F subscript 1 generation. And you can see that there's a little definition here. The F1 generation means the set of offspring that results from a parental cross or a cross. The F stands for the word filial, which means son or daughter. So we have an F1 generation, which is the first lot of offspring from a parental cross. Okay, and you can see that the offspring there is a heterozygous offspring. There is only one possible outcome from this cross, which is the RW pink snapdragons. And of course, each one of these blocks is 25%. And so we can say that there's 100% possibility that all of their offspring is going to be pink in colouring. Okay, now this is our second cross. I've taken both offspring from the F1 generation, and I'm going to cross those pink F1 generation snapdragons together. This is our second round of cross, and we're going to have some offspring which we call the F2 generation. So all of the offspring from the F1 cross are called the F2 generation. So they're 2 generations away from the original parental cross. Okay, now because each of our parents in this round are pink and they have a genotype of RW, each one has a red allele and a white allele to donate. And you can see from the cross-matching here with the arrows, you can see that we have a variety of results for the F2 generation, when our F1 generation is heterozygous pink. So we can see in our Punnett square that I've got parental 1 here, which is a pink colour, and parental 2 is a pink colour as well. And from that cross, we can see that we get an R red and an R red allele for our first 25%, which matches this one here. Okay, so that's going to be a homozygous red snapdragon, just like the parent one in the parental cross. Now we have 25% and 25%, 50% of the F2 generation. There's a probability that 50% of them will be pink with a genotype RW, just like the parents. And then there's also a 25% chance that the F2 generation will be white, just like the parent 2 in the original parental cross. Okay, there are three possible outcomes for the cross. They are RR red, RW pink, and WW white. And I've just put them down here in that table. I think it's a bit cut off here down here for you. So just here in this section it says WW 25%. Here the possible phenotype is white petals, and here the allelic combination is homozygous. Another example, here are Andalusian chickens. We have an intermediate phenotype based on the crossing of 2 homozygous parents, the homozygous black chicken. That C stands for colour, the B stands for black, so colour black, crossed with colour white chicken. And all of those F1 generation offspring will have one of the alleles from both parents. So it would be colour black, colour white. And these F1 generation birds have an intermediate phenotype in between both of those parents, and we call that a blue chicken. Okay, and you can see from that first Punnett square, we've got our black parent, we've got our white parent, and because of that assortment of donation of alleles, we have these blue, all of the offspring are blue. So 100% of those F1 offspring will be blue phenotype. Here, just like with the snapdragons, if we cross the 2 heterozygous F1 offspring, the blues, BW and BW, then we again get the same pattern, 25% matching one of the parents, the other 25% WW matching the other parent. And then we have those 2 intermediate phenotypes again, the heterozygous phenotypes. All right, so here we've got some more cutting off, I think. Possible genotypes will be WW 25%. This section here says white feathers, and then the one that we're missing down the bottom here is homozygous. Okay, our last example here is in humans. I wanted to get an example that was relatable to us. So an example of incomplete dominance in humans is hypercholesterolemia. This is where the body doesn't absorb does not absorb or produce the cell receptor to enable the absorption of LDLs. I think there's a typo in there. To enable the absorption of LDLs, so that's low density lipoproteins, which is a type of cholesterol. You have several different types of cholesterol in your body, and low density lipoproteins are the ones that are not as good for you. So they're still essential to your diet, but too many of them could be quite bad. And the disease here is where the gene will either produce a receptor that sits on the surface of your cells and absorbs the low density lipoprotein from your bloodstream, therefore reducing the concentration of LDLs in your bloodstream, or they don't. In this case, the disease here does not make any of those protein channels in your cell membrane, and so therefore it doesn't actually absorb any of the LDLs in your bloodstream. If they're not absorbed, they build up to really dangerous levels in your bloodstream, and they can attach to your artery walls, causing thickening of your artery walls and just dangerous blockages so that your blood can't pump through them. So the homozygous here, capital H, can make the receptors for the protein, or for the LDL. And here the homozygous, lowercase h, is not able to make those protein channels in the cell. The heterozygous has one of the alleles of both of the parents and can make some of those protein channels. So they have a mild risk of disease here. So the homozygote for the disease will produce no LDL receptors, therefore will not absorb any of the LDLs, and therefore their life expectancy is usually less than 20 years.